So let's begin the presentation sections. So each presentation will be 35 minutes, followed by 5 minutes question and answer sections. So we welcome our first presenter, Professor Nathalie Wong from the Chinese University of Hong Kong. Please, Professor Wong.
I was told to speak down to the speaker. Do I look into the slides here? Do I have my slides up? I don't see my slides here. Can somebody help, please? Thank you very much. Yes. Thank you. Thanks, Dennis. Thank you. Thanks very much. So I'm Nathalie Wong, project coordinator of this theme-based research scheme-funded project on the liver cancer genome. Good morning, and it gives me a great pleasure this morning to present to you on behalf of the group. So what I thought I would do is to introduce to you the team or the institutes that are involved in this project. This is an interdisciplinary team with members of the group coming from the Chinese University of Hong Kong, the University of Hong Kong, the Hong Kong University of Science and Technology. As well, we have on board with us our members from the Beijing Genomics Institute, Shenzhen, and also a few members from the State Key Laboratory of Oncology in South China, and these members are from Guangzhou, our sister laboratory. So before I go into the some of our research progress, what I thought I will do is to give a couple of slides of background on our research directions and objectives and the impact of this research eventually to patients. So to begin with, just liver cancer or hepatocellular carcinoma actually accounts for more than 90% of the annual diagnosis of liver cancer. This is a highly aggressive tumor that is prevalent in China, in Hong Kong, and Southeast Asia. We do see an annual incidence of about 320,000 new patients with HCC per year, of which more than 50% actually occurs in China. In Hong Kong, HCC currently ranks the third leading cause of cancer deaths. The cancer itself is actually associated with very poor survival prospects and prognosis for most patients. And here you can see a near equivalent ratio of incidence to mortality, which is a strong indicator of the poor, dismal clinical outcome for most of our patients. The reason why these patients have a dismal clinical outcome is actually because there are certain problems with the current clinical management of our patients. So here I'm listing the two major aspects. The first is the limitations of the current screening program. It's not very effective as such that really by the time the patient presents the liver cancer or HCC, it's already in its advanced stage. And so the early diagnostic marker is actually much required and is actually essential in order to pick up those early HCCs and offering our patients a surgical option. The second is the low therapeutic efficacy for most of these patients, I think in more than 85% of them. Because these patients are being picked up at the advanced stage, so therapeutics or chemotherapy currently is the one major option that we are offering our patients. However, even with the best FDA-approved drug, the sorafenib, while the Western data is suggesting a median survival of 11th month, our Chinese data actually is only about 6.5 months, really not a lot of improvement here. So to go a little bit further on the limitations of our surveillance program methods at the moment, we are mainly using the alpha-fetoprotein in ultrasound. But the problem with the alpha-fetoprotein of its sensitivity is only about 70%, and there is a false discovery or false positivity during the acute hepatitis and the false negativity in about 50% of the early HCC, which makes that 50% of the HCC cannot be picked up because of its only 50% pickup rate. Ultrasound, on the other hand, is actually labor-intensive, operator-dependent, and the sensitivity is only about 80%. So with that, The ultimate mission of this project is really hoping to understand the disease pathogenesis better so that we can eventually improve the clinical outcome of our patients with HCC. And what we propose to do is to clarify the multifaceted genome alteration that actually underlines the tumor biology of the HCC development. Before I go further, I would like to highlight to you the etiologies associated with HCC. Chronic infection with the hepatitis B, HBV, is a strong predisposing risk factor for the development of liver cirrhosis and HCC. It is believed that chronic carriers of the Hep B has an increased risk of 100 to 200 times more than those non-carriers. So Hep B is a strong predisposing factor. Here shows you the global incidence of the HBV infection, where you can see there is really a very high incidence of HBV infection in China. And it is not surprising that then in our HCC patients, more than 80% of them actually are arising from the chronic infection of this virus. So to give you an idea about Hep B infection globally and the incidence of the HCC, here highlights to you, globally at the moment we have about 400 million infected individuals with the Hep B, of which these will be considered as the high-risk population. And then about 100 million of them will develop into acute hepatitis or the active hepatitis. From this population, annually, about 4% of them will develop of these who subsequently will develop cirrhosis. About 4% of them will develop into HCC, which goes to the figure that I showed you is about 320,000 per year, of which 50% is occur within China. It's becoming apparent these days that the epidemiology of HCC is evolving. So more recently, we are seeing that metabolic syndrome has emerged as a significant cause of HCC. And by metabolic syndrome, one is referring to a group of risk factors, which includes obesity, diabetes, hypertension, and dyslipidemia. The non-alcoholic fatty liver disease, in short NAFLD, is considered the liver manifestation of the metabolic syndrome. And once the patient has this NAFLD, it can actually progress more, advance to a more aggressive form of the fatty liver disease termed as the non-alcoholic steatohepatitis, in short, NASH. In fact, NASH is becoming increasingly recognized as a serious pathological condition that causes the progressive hepatic damage, cirrhosis, and eventually HCC. So the occurrence of this NAFLD in our population is about 700 million, arising, as said, mainly from the hyperlipidemia, diabetes, hypertension, and so on. And then this is considered the high-risk population, of which 200 million will develop into NASH. And because NASH has a strong association with cirrhosis development, about 3% per year of these NASH-induced cirrhotic patients will develop into HCC. We are seeing currently about 50,000 HCC arising from these patients per year, and the trend is increasing. And it is believed that NAFLD is an important public health problem worldwide and increasingly prevalent in China as well. So, although in the NASH-associated HCC, it would seem like only a very small percentage from the NAFLD big population actually is considered to further develop into NASH. But because NASH is so closely associated with the development of cirrhosis, this actually is an alarming number eventually. And moreover, diabetes, obesity, and hyperlipidemia is becoming increasingly prevalent in this world. So such a big denominator of NAFLD will invariably translate into a big number eventually and in due course. So despite there is a causative link between NASH and HCC development, currently there is really minimal information related to the genetic alterations associated with the NASH-induced HCC. So we know that now progression of liver carcinogenesis involve two major etiologies in our locality. So that's hepatitis B and NASH, of which both is very prone to the development of liver cirrhosis. And from cirrhosis, we know there are patients who will eventually develop into HCC. And it is this transformation from liver cirrhosis to HCC that often information is minimal. So in this project, we aim to understand the HCC pathogenesis, and to advance our basic knowledge in this aspect, we propose to comprehensively uncover those tumor-associated genetic events. And in order for us to compile or construct a genetic blueprint and give us the genomic landscape of these HCC, we are, in this project, integrating information such as the single nucleotide variation, which you can look upon like mutations, copy number variations, which is the gene copy gain or copy lost. Structural variation, in another way to look at it, is the rearrangement, genome rearrangement, chromosomal rearrangements. Transcript level will give us information on the gene, the expression being upregulated or downregulated, and also the clinical data. Together, we hope to construct a blueprint, a landscape of each case of our patient, and then we will have a better, good understanding on the background of the disease causation. So in order for us to achieve that, we are using the technology called the next generation sequencing, or also termed as the massive parallel sequencing. This kind of sequencing work allows the unprecedented depth and speed in the genome sequencing, and at the same time can give us information on the copy number and the structural variations. The NGS sequencing, or the massive parallel sequencing, already is providing the field with enormous potentials in terms of gene discovery, and those we now know that has actually provided us with a lot of information or genes that allowed us now to predict disease onset, severity, and response to therapy. And just to very briefly, just 2 slides on the principle of this massive parallel sequencing on this chip with lanes where we can actually seed the PCR product from the library construct of the tissue. We allow them to anchor to the chips, and then further allowing it to expand to become clusters, and thereof sequencing would activate. And because each of these PCR products are allowed to further amplify on the chip itself, we are actually seeing much more sequence reads per case. And to give you an idea how advanced is this technology, anything in the currently still in use will be the Sanger sequencing, and that involves in the cloning or direct sequencing, and then also if you look at the capillary electrophoresis reads, we're talking about 1 read per capillary. Now, however, with the NGS sequencing, what we're seeing here, because these are real-time sequencing and then they have got clusters on these lanes, then we are in a way seeing that almost like more than 1 million reads per array, and the data generated between you can already appreciate it's a colossal amount of information generated from just one sequencing reaction. So with all this amount of sequencing coming out, bioinformatics is an indispensable aspect of our project. So we have developed over the last 2 years genome analysis workflow and transcriptome workflow. So very briefly will be that we, from the generated raw reads, will have to filter, clean them, and then also align them to the reference genome. And then after the alignment, we will start analyzing them for the somatic mutations, for the copy number, and for structural variations, and so on. Eventually, what we hope to achieve is we have some information already, and it's those genes that has actually significant implication in the carcinogenesis, and then more importantly is our experimental validation that will actually substantiate our bioinformatic analysis. The same with the transcriptome analysis is being adopted, where we will clean the raw reads, map them, and look for information, as I've mentioned earlier. And then again, we have to prioritize our analysis because the information is a lot. And this prioritization in terms of the, say for instance, will incorporate the gene analysis, the copy number, and the gene expression. And then finally, again, will be the expression, the validation analysis, which is indispensable in this project. So this gives you just one example of the kind of information that we will get. If you look at the somatic substitutions, we can get something like 80,000 to 100,000 these kind of SNVs, and it will cover both coding, non-coding, intronic and intergenic regions, and then just like opening a full map on the kind of mutations that is present in per case. Looking at it in another way is that we see that already within the intronic region that the mutations is the most abundant. Many of us will be interested in the coding region, and you can see here, so this example, that we will see synonymous, the silent mutations, and also the non-synonymous, which will include the missense and the nonsense mutations as well. So itself, it's a very, very powerful technique that gives us a lot of information in one reaction. We have already done a good number, sizable number of Hep B associated HCC and NASH associated HCC, and then we have begun to actually analyze the genome pattern to see if we can see any distinct differences in terms of the overall mutations. And straight away, looking at this one, which is the telling us about the transitions or the transversion mutations, we are seeing there's actually more transitions than transversion. And we, in particular here, the A to G transition has been reported, not only by, not being seen by us, but also reported by other HCC groups to suggest that it's also associated with other etiologies like the Hep C as well. And it would seem that it's a characteristic mutation changes of the HCC genome. Another transition is seen as the C to T. This one has actually been reported in a number of cancers, now also in HCC, and it would seem to be to do with the deaminases activity. We then went further and looked deeper into the exonic mutations, and here gives you the idea on the transitions and transversions again. Between the Hep B and the NASH, we already you can see spontaneously the C to T transition seems to be quite high in both the NASH and the Hep B associated tumors. And this kind of deamination of cytosine has been proposed in other cancer types to be due to the upregulations of the cytosine deaminase family, the APOPAC, and we are also seeing the APOPAC family members being upregulated in our tumors as well. So maybe that this is one of the links associated. Another point is what we noticed between the Hep B and the NASH is that we are seeing the transversion between the T to A seems to be quite high in the NASH-associated tumor compared to the Hep B. And when we looked into the large database, this was reported in the science paper of more than 70,000 cancer samples, we are not seeing this kind of mutation in those cases. We don't know what is the reason behind of this transversion being high in the NASH patients, but it is possible that this kind of a T to A transversion has been reported to be a signature of this drug AA. In Chinese, it's called the which is actually a natural product found in many of our Chinese herbal remedy. And in this kind of remedy is being used in weight loss, in going on a diet, or in relieving the menstrual pain and so on. So it's not sure sort of what is the underlying reason, but that could be one possibility. Besides looking at the genome globally, we have also started looking at the genes in particular, and especially those that may be associated with the liver carcinogenesis. So one of the genes that we picked up is the beta-catenin mutation, which seem to be clustered within the exon 3 and with some sort of found in the exon 8. An interesting note to see here is that the beta-catenin mutation seems to be more prevalent in the NASH-associated HCC than the Hep B. So it may be an important sort of driver event in the NASH carcinogenesis. This exon 3 is actually the region where the GSK phosphorylates the beta-catenin. So by having this mutation, it will invariably inhibit the phosphorylation and therefore the degradation process of the beta-catenin, and allowing the beta-catenin then to translocate to the nucleus where the transactivity occurs. We actually cloned each and every single mutation that we found from the sequencing, and we did the transactivity assay, which showed increased, and also in the growth assay, which you can see here that distinctively the ones with the mutations actually showed increased colony growth compared to the vector. So hence this oncogenic mutation, we can refer to that as now, it actually enhances growth and enhances the activity as well. As I said just now, that it seems that the beta-catenin mutation is actually found more in the NASH-associated HCC, and we can actually recapitulate that in an animal model. In this DB mouse model, we see that in a couple of tumors that were induced from the den, there were actually seen the beta-catenin mutation. So we're pleased to see that the actual carcinogenesis can be recapitulated in an animal model, And this animal model actually is now quite handy sort of model for us to work on the therapeutics. The ultimate goal of our project actually is from cancer genome to start looking for targeted therapy. And with that, what we aim to do is to develop personalized medicine where the information of a patient genome can be used to select for them the most appropriate therapy. And often, these will be important biological factors that are linked to the cancer cell growth, survival, and these will be those specific genes or those genes that are those proteins that are encoded from these specific genes. What we hope to eventually develop is a pipeline where patient's tissue, be it biopsy or surgical tissue, will be subjected to histological examination, and from there we will provide the patient with a battery of analysis, which will include the fluorescent in situ hybridization, where will give us information on the copy gain and also in translocation, and then the protein encoded, the expression, okay, shown here with the immunohistochemistry, and last but not the least, of course, will be the sequencing to confirm that the patient indeed has the mutation. All this information, we will group them together and then subject them to analysis, and in order to define those specific genetic variants, and thereof to provide our patient a very personalized treatment. We have already, I apologize for this very, very busy slide, but I will try to explain. We have already embarked on the analysis of a panel of the druggable targets. And here on top, these are the genes, and this part are the specific mutations that we have picked up on these genes. Down here, these different squares actually represent those mutations that we have picked up. The pink ones represent those tolerated mutations, the darker color represent those deleterious mutations, both from prediction software. So we have included them in our analysis because we don't know if those tolerated ones eventually can be also druggable or not. And then more importantly is that many of these mutations actually fall within the conserved regions of these genes. So once a mutation falls into a conserved region, it is more likely highlighting an activity that will affect the activity and hence more druggable in that sense. From our analysis, we already picked up one potential candidate. This is the JAK mutation, which is found in about 5% of our patients. The percentage incidence may be still low, but at least from the oncologist's point of view, there is a target that we can start on and work on. So here just gives you the example of the mutations that we have picked up in 2 tumors, and it is absolutely absent in the adjacent non-tumoral liver. During our analysis, we have actually picked up an interesting target, which is the HTAC histone deacetylase 7. The interest of this gene or protein is that all the mutations, or predominantly the mutation, actually clustered within a particular region of the active deacetylase domain. So we are very excited now with this finding of late, and we are currently working further on the potentials of drugging this, and then also substantiating the role of this gene in patients' therapeutics. We actually are equipped within our group also the ability to do some protein structure prediction. So taking HTAC as an example, what we have begun to do is that to understand if these kind of mutation actually mean anything to the activity or the binding of the protein. So giving you an example would be somewhere down here where glycine is mutated to glutamic acid. And this is an interesting one because it actually, the glutamic acid has got a higher polarity than glycine, and that is predicted to affect the actual binding of the enzyme to the motif, and that may reduce the activity of this protein. So that seems to be quite interesting for these mutations that we have picked up. We now know actually many different cancers, and we hope to actually achieve this in liver cancer, is that cancer patients with specific gene mutation can be classified into different subpopulations where we can offer these patients with therapeutic implementations. We hope to eventually achieve will be from our cancer patient, the HCC patient, to select those with a particular mutation and offer them targeted therapy, while the others will offer them alternate therapy for the time being. by doing so, what we hope to achieve is to provide our patient with the molecular target or the predictive target, a good prognosis, and then actually, even with these unfavorable prognosis one, we hope to find continuously other kinds of marker that will improve the clinical outcome of them. And I think it goes without saying, to achieve all these goals within this project, it really needs a big team. Team that not only is from different discipline, but also, more importantly, is the interdisciplinary collaborations and activity that is essential. And I'm very pleased to say that we have this kind of activity within our group. So within our team, our project, we have got clinicians, basic scientists, and also people with expertise in cancer genome sequencing, and bioinformaticians. Our clinicians are all frontline. They are actively engaged in the liver management or HCC disease, hepatologists, pathologists, oncologists, and surgeons. And they are also not only involved in clinical trials, but also the translational research. The basic researchers, many of my team members are experts in the fields of signaling pathways, genomics, NASH biology, and animal modeling, which are indispensable in this research. And together we do integrate, and we do talk amongst ourselves into achieving what is best for the outcome of this project. So what I hope I have achieved in the past minutes or so is that I'm a strong believer genome analysis is very important, and I hope I've conveyed this message to you, that such analysis is important to underpin the molecular basis of the tumor biology, and thereof it has great influence in the medical progresses and the promote of the development of targeted therapy. What ultimately we hope to do is, from the cancer genomics, reaching out to our patient and providing them with personalized medicine. That's the end of my presentation. Thank you very much.
Thank you, Professor Wong. So any question for Professor Wong from the audience?
So what is the sample size supporting the enrichment analysis?
At the moment, we have done 80. We have done 80 tumors. So we are going to push it up to 100 something, and then have a look and analyze them for prognosis or therapeutic potentials. And we have also cell lines harboring those mutations, so ready resource for us to test out the new drugs.
So Nathalie, thank you for a very nice presentation. So I have a very simple question. You were showing the demographics earlier. I noticed Canada is all red in North America,
Alaska.
whereas the
That's Alaska. That's Alaska.
I mean the Alaska area.
Yeah, Alaska.
Yeah.
More
So whereas the rest of the
US
so any particular reason? Just curious.
I think somebody asked that question before. We don't really have a good answer to that, but maybe it's to do with the people migration. Say some people, I mean the population migration in sort of the earlier days that actually migrated with the Hep B, and so they may have gone to Alaska.
Oh. Okay, so that must be sort of
evolutionary
I
development.
see.
Yes.
I see.
So Hep B have been with us for a long, long time.
I see. Okay. Yeah, just curious. Because the rest of North America looks pretty decent.
Alaskans have Hep B. They have HCC as well.
Yeah. Okay. Thanks.
So any more questions from the audience?
Please.
So I'm not so clear about the beta-catenin and the animal model. Are they related or
So it was just this animal model is a NASH model, which will develop HCC after the chemical carcinogen induction. And it was very interesting to see that there is beta-catenin within that, which falls with the human results. So it seems like the animal model has recapitulated the human results. So hence we were quite excited that now we have a model that we can work on. On top of that, We are now developing a transgenic model, truly inducing the animal with a particular kind of oncogenic mutation, crossing it with a phosphorus background or DBDB background, as I have shown, and then we're going to observe if that has actually an advantage in the HCC development without giving it chemical carcinogen, just feeding it with fat.
So what is the function of exon 3?
Okay, it's the GSK, this phosphorylation domain. So by having this mutation, actually, because GSK degrades GSK degrades beta-catenin. So by having this mutation, it blocks the degradation, and so beta-catenin is remain active
So
in
it
that.
cannot be regulated.
Yeah.
I see. Yes.
Hi, very nice talk. I have a quick question for you with the sequencing and transcriptomic components. Was someone else speaking?
Yes.
Okay, sorry. And so when you're doing these analyses overall, are you basically sequencing samples which are from the cancer patients or the disease patients, and you have controls as well such that these are very good as far as prognostication or reactive due to the disease process and the other changes that are going due to the overall somatic gene mutation rate that can occur?
Yeah. The study design that we are using now is every single tissue or every single patient will have the tumor and a normal. And then for that we will do the genome and transcriptome paired. So you can imagine from the genome we'll have the full set of the tumor and tumor adjacent, and then also from the transcriptome tumor and tumor adjacent. So then this is really HCC patient, and then it has a control in it as well.
So thank you again, Nathalie, for your talk. I want to go back to the animal model, which I thought was really interesting in the sense that you're having to give a second hit to see the disease occurring. I mean, you don't think the second hit in your patients is actually a carcinogen? You're suggesting it might be dietary or what?
So with the DBDB mouse, there are certain limitations because it needs that push. Otherwise, it will actually take. It may or may not develop liver cancer. So we need with the DBDB background to use a DAN chemical carcinogen. There's another animal model which we are now turning to is the phosphorus, which with that is the you feed it with the high-fat diet, it will gradually develop steatosis. And all the pathology that just mimic the NASH, it's all there in the phosphorus, and then gradually it will develop into HCC in some of them. So, but that takes a long time. So now what we want to do is a transgenic cross between phosphorus and beta-catenin and see if that will actually enhance it.
Thank you.
Very interesting talk. My question would be in terms of early detection to some of these patterns of behavior, and you highlight some very alarming rate of that by the time you provide detection, it's quite mature. Has there been any experiment done in looking at some of the confirmed cases and backtracking and developing historical data set to develop computational model, which could then allow us to make prediction at a much early stage?
Okay. You're asking if we can backtrack into the tissue and look for the aberrations earlier on. So I think this is actually a part of our project as well, if we can do that. We have a surveillance program within our hospital that we have the happy infected patients that comes in that we have biopsies from time to time, time intervals. So some of these patients actually developed into liver cancer eventually. So then what we hope to do is that based on the information of the genome is that we're going to see if it's in the HCC tumor, and then if it's in there, then can we backtrack into the earlier biopsies. And because biopsies, are very, very small material, very precious, so we are going to wait until we have really some solid data before we use those tissue samples. But yes, we will be doing that as well.
Sorry to ask you another question. It's more like a suggestion. So was the earlier question about beta-catenin piqued my interest. So if the mutation ends up in an alteration in the steady state of concentration or level of beta-catenin, then it raises a lot of other interesting possibilities, and you probably know that beta-catenin is one of the components in exosomes that are shed by cells who want to downregulate when signaling as quickly as possible. So instead of going through all this phosphorylation, blah, 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 and they just throw it out. And then it's picked up by, most likely picked up by some other cell, and then therefore starting something else. So I know you're interested in exosomes as well. So I mean, I think these things can be linked.
Yes.
Actually, that the cancer cell that harbors the mutation can then ends up with an unusual load of beta-catenin, and if it goes the dumping out the garbage type of mechanism, then it can influence the microenvironment, and then et cetera, et cetera, et cetera. So
Okay.
anyway, just a thought.
Thank you very much. I
Yeah.
shall integrate between the two projects.
So if no more questions, so please thank Professor Wong.
Thank you.